Forensics uses physics, chemistry, biology, and other sciences to analyze evidence recovered from crime scenes. Things like fingerprints, trace chemicals, bullet casings. About 30 years ago, DNA testing was added to the forensics toolkit, replacing blood typing as the primary method of identification. All biological samples contain DNA. Skin, hair samples, blood, and other fluids. Even tiny amounts or partially degraded samples can be recovered. This makes DNA testing ideal for use in crime settings. Although 99% of DNA sequence is identical from person to person, there are regions of the genome where the chemical sequence varies. DNA testing takes advantage of these differences, looking at about 20 of these regions across the genome and compiling the information into what's called a DNA profile. Now, these DNA profiles are also used to establish family relationships like paternity and maternity testing. Each DNA profile is unique, just like a fingerprint, and can be used to identify an unknown person, to place an individual at the scene of a crime, or to remove a suspect from consideration. The markers that are used in a DNA profile are in regions known as non-coding DNA. That is, sections of the genome that don't provide direct instructions for making proteins. Therefore, they can't be used by investigators to understand the physical characteristics of the individual that's left the sample behind. A new set of molecular markers has come to market that does provide this kind of information, giving insight into physical characteristics like eye and hair color, skin tone, ancestry, even general facial shape. This new approach promises to create the genetic equivalent of the artist's sketch. In this session, we'll explore more about molecular fingerprinting and ask whether a tiny sample of DNA left behind at a crime scene really can be used to create a realistic and accurate representation of its owner, an image that would be recognized by others. Good evening. Welcome to the 2018 edition of Biotech 201. How can Biotech? It is great to see you here this evening, those of you here in the Jackson Center, and those of you that are watching from home. We are really glad to have you with us on this February night. We have a lot of ground to cover. We have a whole lot of stories that we're going to talk through over the next 4 weeks. And we're going to focus on a set of questions that I'm often asked as I'm in the public. So how can biotech do this or do that? Or I saw and read this, or this was in the newspaper. So these are stories that are essentially curated from questions that you and other members of the public ask, plus a few other pieces that I've thrown in just because I think they're really cool and they're a great example of what genetics and biotechnology can do. DNA forensics is one of a whole host of things that are used to try to identify evidence left behind in crime scenes. And that starts with things like fingerprinting, which was first developed around the turn of in the early 1900s, the late 1800s. Things like bite mark analysis, blood splatter analysis, all the sorts of things that we think about when we hear concepts of forensics. The challenge with many of those other pieces is that how well validated they are, or how well validated they aren't. Hair analysis, bite analysis, blood splatter, all have been used for years as ways to evaluate evidence in a forensics and a criminal case. But we've recently begun to realize that some of the evidence, some of the ways we were interpreting the evidence varied greatly from one individual to another. So there's been a lot of questions recently about the use of those kinds of tools in making decisions about evidence, and there have been a number of criminal cases that have even been brought back up or even overturned because of the way the evidence was analyzed and not necessarily standardized from place to place. That's not the case when we talk about DNA forensic interpretation. And that's potentially because DNA analysis was first developed in the lab in a scientific setting where it was thoroughly vetted before it was brought into the field of criminology. So that's why when we talk about DNA testing in a forensic setting, it's often called the gold standard. And it works off segments of your DNA that are called STRs, short tandem repeats. Let me give you an example of that. So here's a stretch of your DNA, and it's kind of hard to notice unless you really look carefully, but here is a repeat. And you can see that this repeating sequence is present 6 times in this stretch of DNA. All right, so this is a short tandem repeat, and it's repeated 6 times. Now, you've actually got 2 copies of all of your DNA, a copy you inherited from mom and a copy you inherited from dad. So, for example, if you've got 6 repeats on one chromosome, you might have 4 on the other. The concept of STRs is that they were specifically chosen in forensic testing because they vary greatly from person to person and across populations. And so this individual, if this was what they had at this specific STR, this specific site in their genome, they'd be tagged as a 4-6. 4 repeats on one copy, 6 repeats on another. That makes sense? Everybody with me on that? Okay. So this concept, STRs, are used in DNA forensic testing. We actually look at about 20 sites when we're doing forensic testing from a crime scene. We used to do 13. About a year ago, that was expanded to 20 different sites. As I said in the video, these are in what are called non-coding regions. So these are mostly not in genes. They're in areas outside of genes. And even the few that are in genes are not in the part of genes that provide instructions. So you cannot get any information about an individual, any kind of information about disease, about traits, nothing from these STRs. This is just a way to get some identity. So we look at 20 of them, and I'm going to show you. So here's the DNA that's left behind at a crime sample, and then let's just imagine that we've got 2 different suspects. So when we do this testing, when we look at these 20 STRs, we get this whole set of markers. So 8 and 12, 11 and 16, 4 and 14. There's 2 of these at every site. Each one of these is one of those STR sites scattered throughout your genome. And you've got 2 alleles, because you've got one from mom and one from dad. Sometimes they're different. Sometimes, just by chance, the copy you've got from mom and the copy you've got from dad are identical. So in some cases, you may see the same number. So here's the crime sample, and we're going to look at the same markers from suspect 1 and suspect 2. And I think you can see pretty easily on the handout that, all right, suspect 1 has the same combination, and so does suspect 2 as the crime sample. But then as we start looking at other markers, suspect 1 does not have the exact same markers that are left behind. Suspect 2 does. So what that means is we can eliminate suspect 1. Suspect 1 can be eliminated from this panel. I can't eliminate suspect 2. And in fact, that's a really important piece to understand in forensic DNA. I can never say this is the same DNA of this suspect. I can say the likelihood that it was left by anyone other than this suspect is incredibly small, like 1 in 5 billion. So it's an exclusionary. I can definitely say with this kind of data that it doesn't match suspect number 1. I can exclude suspect 1, and I can say there's a very, very small likelihood that I could exclude suspect 2. So the way forensics is done, I'm looking at 20 different markers, and I'm looking to see if I'm seeing the same types of repeats, alleles, in my sample left behind as what I'm looking for in my suspect. A variation of this data is the same kind of thing that's used when we're looking at paternity. When we ask, does this child, for example, which of these 2 men is more likely to be the father, or which of these 2 women is more likely to be the mother? We're looking at the same kind of set of markers, and we are looking to see if the child has inherited a set of variations, a set of alleles that consistently could come from this individual or from this individual. So this is the backbone of DNA forensics, and it's been used since the late 70s, early 80s. It was first developed in England and then brought over to America, and it's been used to solve hundreds and hundreds of crimes by using DNA evidence. Now, generally, you don't use DNA evidence alone. Generally, you're using it in combination with other information. And when you go through and you do these 20 different STRs, you then build what's called a profile, a DNA profile. And that DNA profile is then compared against a database. And this database is called the CODIS, Combined DNA Index System. This is America's DNA database. And it actually is multiple databases dealing with missing persons, convicted offenders, and forensic samples. So this database contains samples where we know where they've come from. This is a body that was identified. This is an individual that's already been convicted of another crime. This is a sample that was left behind at a crime scene. And so when I get my own DNA from this specific crime scene and I run those markers, and I get that profile, I'm looking to see if there's a match between my sample at my crime scene and anything that might be in the CODIS database. And the CODIS database is actually quite large. As of the end of last year, there were over 13 million offender profiles. These are individuals that have been convicted of violent crimes, and their DNA goes in the database. It includes almost 3 million arrestees. So in certain states, if you are arrested for certain types of crimes, whether you're convicted or not, your DNA is taken and the profile is put in. That's somewhat controversial. Different states have made different decisions about that. And it's aided, I don't know if you can see down here at the bottom, it's aided in over 350,000 investigations. So you've probably heard about DNA testing in forensics for a long time. Courts have become much, much more comfortable with it. It's actually called the CSI effect. 15 years ago, before CSI was on television, people were like, DNA testing, what is that? But it is so commonplace now in all of the courtroom dramas that juries who have seen this on television are like, oh yeah, that's good science. That match, oh yeah, that means something. It's a really interesting example of how pop culture has actually changed our perspective on something that's really very scientific and quite technical. So lots of cases, as I said, have been solved using the CODIS database and DNA forensics. It's also been used on the flip side when individuals were convicted and DNA evidence was available, and the DNA evidence could be tested sometimes years later to show that they could not have been the one that committed the crime. So there are a number of groups out there. One of them is Barry Scheck's Innocence Project, and they look at specific cases where they think that there's enough evidence that it makes sense to have DNA tested, and in over 200 cases they've actually overturned convictions where people were wrongfully jailed because of either eyewitness or because of other issues, and these individuals actually were not the ones that committed the crime. All right, now there are some weaknesses with this type of forensic testing. So even though this is used really as the gold standard, it actually has a significant number of problems. It does not do well when there are tiny samples, when there are tiny amounts. So one of the newer tools, newer technologies, is using what's called touch or trace DNA. So if I touch this surface, I've left behind a tiny amount of DNA from my skin. And I could actually swab that and get a little bit. It's a tiny amount of DNA, but I could actually get touch DNA. This kind of technology does not work so well with minute samples. It also doesn't work so well with degraded samples. So samples that, let's say blood samples that were in a moist, warm environment. So the DNA has been broken down by bacteria, for example. You can do some work with degraded samples, but you've got some real challenges associated with working with degraded samples. It also does not work well when you've got what are called multiple samples. So if you have a sample that has the DNA of multiple individuals, so mixtures, It's very difficult to be able to separate out the individual profiles of all the DNA that was put together. This gets to a pretty controversial point because there are a number of labs that have developed tools that they claim allow you to separate out mixtures. And people pay large amounts of money for this kind of analysis to separate out these mixtures. The companies that have developed those technologies hold them as proprietary. So they aren't available, the underlying algorithms aren't made available to the defense or to the jury. They're just told, essentially, trust us, we've used our own internal analysis, and this is actually true. That's really challenging, and there are a number of groups that have now started to sue to ask for the companies to say, show us what your underlying algorithm is, because if you're going to use that to convict my client, we have a right to know if the tools that you're using are actually appropriate. And then this is the really important part, which is why the next thing I'm going to talk about potentially becomes so important. Forensic DNA, as it currently stands, those 20 STRs, can only be used to make an identification of somebody that is already in the database. So you have to have a match with something that's already been analyzed. If the individual isn't already in the database, I've got to go out and gather samples and hope that I actually am lucky enough to get their sample. So in some cases, this is really valuable. I've already told you it's used in over 350,000 investigations. But it only works if I can make a match in the existing database. So that brings us to the whole concept of DNA phenotyping. Let me try this one more time. Those of you that are watching at home, you actually are lucky tonight because you don't have anything that's keeping you from seeing the screen. While those of us sitting in the Jackson Center, there we go, have been looking at blank screens. All right, DNA phenotyping. DNA phenotyping actually uses genetic information that tells us something about the owner of that information. So it looks at DNA that provides instructions about the physical characteristics, the external characteristics of the individual that left that sample behind. So it could tell us, for example, about the ancestry of the individual that left this sample behind. It can tell us information about their likely eye color, hair color, skin tone, even potentially their facial shape. And some of this information actually is based on data of two of HudsonAlpha scientists, Drs. Greg Barsh and Devin Absher, doing some of this work in understanding genetic variation that impacts like facial shape, the positioning of the nose, how far apart the eyes are, how deep set the eyes are, where the cheekbones lie. So you can use this information, theoretically, to actually get a genetic prediction of the appearance of the person that left that DNA sample behind. Now, we have a lot of good knowledge of the science, the genetics behind pigmentation, skin, eye, and hair color. We can predict those pretty accurately. When we start looking at things like height, how likely you are, if you're a man, to have lost your hair, to be going bald, your age, if your hair is thick, fine, curly, and your general face structure, the further down this list we go, the less likely we are, the less amount of information we have to be able to make really good predictions. So you got to take this with a pretty large grain of salt. But the more we learn scientifically, the more we add to this, you can begin to actually build what's called a biological witness, which can be as powerful, as valuable as an artist's sketch. So there's a great article in National Geographic from July of 2016 that I would encourage you to take a look at that talks about how you take that same DNA sample, and in addition to running the 20 STR markers for the CODIS profile, You actually then look at hundreds of other markers across the genome, markers that you know are associated with these externally visible characteristics. And then there's a whole set of sophisticated algorithms that say, well, if you've got this combination and these over here, then your facial shape is likely to be more or less like this. And your eye color is likely to be more in the blue range or more in the brown range. So you build a whole set of predictions. This actually looks like a photograph. It is a genetic prediction. So this is an image based on the bits of information we know that we can predict from your genetics. And then we can, artists can take that information and they can make modifications. They can, for example, as you can see here, they can age an individual, age this profile. They can change the weight of this profile, so they can begin to get a sense of what the individual that left this sample might look like. Now you're going to hear me say might an awful lot, because it isn't a guarantee. There are a whole lot of pieces we don't know. There was a really famous issue of an artist about 3 or 4 years ago when this technology was really first coming out that walked through Central Park in New York and picked up leftover food that people had discarded and swabbed the food and got DNA from the people that had eaten the food, and then ran these tests. And this artist then built masks of what she thought the people looked like and hung them in art galleries. So there are all kinds of issues associated with this, but samples that you leave behind, either because you threw the half-eaten apple on the ground or because you cut yourself in the middle of a bank robbery and left some blood, those samples, discarded, leftover, disposable samples, are free game, at least from a forensics perspective, whether that's true from an artist's perspective or not. But still, that concept, same kind of concept. So there are a number of companies that now offer this as a service. One of them is called Parana Nanolabs. They've been in the news quite a lot because a number of cases, cold cases, have now been solved because officers turn to this kind of technology. And so again, they take the DNA, they look at thousands of different samples, they run through pretty sophisticated algorithms, and again, those algorithms are proprietary, so if you wanted to know how they made these determinations, you won't get that information. And then they come up with a prediction. So this is, for example, a snapshot. They call this the snapshot profile. You can pay somewhere in the neighborhood of $3,000 to $5,000 to have a snapshot profile crafted, depending on the condition of the DNA that is left behind to work with. And you can see here, they can give you a sense of what the prediction of the skin color is, and what's the prediction of the eye color and hair color and freckles and facial shape. This particular image I'm showing you because it also shows you the photo of the actual individual that was arrested, that ultimately committed the crime, that left behind the DNA. Just to give you a sense of the comparison. So again, this is genetic prediction of what an individual looks like based on an incomplete set of data. The owners of the company say, this is an important tool, not necessarily to say we need to be looking for someone who looks exactly like this, but to say we can exclude, or we can likely exclude suspects who are significantly different from this. So they suggest that it's used as a tool to help officers focus in on sets of individuals instead of saying, we're looking for someone that looks exactly like this. So there have been a number of crimes that have been solved with this kind of tool and technology. But it is by no means a perfect set of technologies. So let me stop and see if we've got questions before I move us a little bit further on in this story. Francis. Who has access to CODIS? Let's see. CODIS, I know that state and federal officials have access to CODIS. I'm not sure if you just like actually, I know, for example, that you couldn't just go type in CODIS and go to the website. I think maybe you actually, your FBI, your local crime lab, your DNA forensics testing lab comes up with this information, determines the profile, and then they query the CODIS database, and then they can tell you if there was a match or not. So it isn't something that you can like DIY analyze. There's a limited access to it. Ooh, if somebody joins the military, is their DNA taken? So I know that the military has a separate program where they take your DNA and they hold it in case they ever unfortunately need to identify a set of remains as being yours. Francis, I don't think it goes in. I'm pretty sure it does not go in CODIS, but I'll confirm that for you.
Do other countries have a similar CODIS?
Do other countries have a similar CODIS? Other countries do have a similar database, and there is the ability to actually cross-check databases in other countries as well. Can they DNA at birth? Yeah, they sure can. I mean, you can do this with any DNA sample. There are some ethical challenges around putting somebody into this kind of identification database when you have no forensic reason to do so. There are certainly those that have advocated that this should be developed, but if you're going to put them in the database under the presumption that they may commit a crime later in life, that's a little bit challenging.
Does the match have to be 100% typically, or would you ever see, well, it's really very, very close, there might be one or two?
Yeah, does the match have to be 100%? There's a little bit of leeway that's allowed. You've got to get pretty close to 100%, but there are some biological reasons why you might not be exactly at 100%. You might have one allele that's a little bit different. But generally, it's got to be pretty significantly close. And there are some really interesting examples of people that have tried to game the system. An individual that was actually a plastic surgeon that sewed into his arm a vial of someone else's blood, so that when they drew blood, they actually drew this other individual's blood so that it did not match. I mean, that's pretty, somebody has some knowledge there. And the other thing that you might be thinking about is, what about identical twins? You have the exact same genetic information as your identical twin. So if you have an identical twin and you commit a crime, it genetically would be very, very difficult to determine if it was you or your identical twin that committed the crime. Not that I'm advocating that anyone in this room who is an identical twin go out and start a life of crime, but that is a challenge to really get at.
How are the exclusion probabilities derived?
How are the exclusion probabilities derived? So we're now going to be at the edge of my knowledge, but you are looking at the frequency of each of these numbers of repeats within a given ethnic population. So you can say, within this population, let's say of Caucasians, how frequently do I see 6 repeats? How frequently do I see 4 repeats? And then when you do that over enough different, when you do those frequencies over enough different markers, you're able to come up with a vanishingly small likelihood that anyone else on the planet would have the exact same combination of markers based on what you know about that population. But that requires that you have a large enough reference sample for each of those different ethnic groups. And if you don't have large reference samples for certain ethnic groups, you cannot use the data from another ethnic group. The results are going to be different. And that's, historically, our DNA data sets are really Caucasian. There are lots and lots of Caucasian representation, and there's not been much representation from any other ethnic group. And so there's been a real push, not just in forensics, but in everything where genetics is important, to try to get more diversity so we can get more information. I'm going to come right here and then right back there.
So this gets to the 1 in 5 billion probability or whatever it is, with the exception of identical twins.
With the exception of, yes.
So if my profile is defined by 20 data points, each consisting of 2 numbers, must vary an awful lot. I mean, what kind of numbers are we talking about as far as?
Yeah, those repeats do vary a great deal. So you may have 20 or 30 or 40 different options within the population at each one of these different markers, at each one of these different pieces. I can dig into the science behind this more and give you guys a reference if you want to actually see examples of how we get to that 1 in 5 billion kind of number.
I take it that the way you have the small sample, you have to amplify the DNA before making the matches.
Yes.
What is the chance that the amplification introduces error in the number of?
So when you have a very small sample, when you have any sample, in order to get enough to do the analysis, you actually have to amplify the DNA. So you use a process called PCR, which is like an electronic copy machine. And PCR, excuse me, can introduce errors. And if you're sloppy in the lab, you can introduce errors. You can amplify the lab technician's own DNA as well. So you've got to be really careful about the way you handle the sample from the moment it's identified all the way through how it's gathered and amplified and analyzed. That's part of the challenge where minute samples come in. Because if I'm trying to amplify a tiny, tiny, tiny sample, I am also potentially amplifying error as well. And that's where some of the analytical challenges happen. Okay, so let me move us on. Here are some limitations of DNA phenotyping, of this being able to identify these external characteristics. You and I change our appearance all the time. We dye our hair, we grow facial hair, or we shave off facial hair, we put colored contacts in, we apply a bronzer, or we go visit the tanning booth, we have plastic surgery. We've just come out of the holidays, and so some of us actually have a little bit more weight than we started the holidays. We do things that change and alter our appearance that are not going to be detected by your DNA. Your DNA profile can't say anything about this person has had plastic surgery, this person wears colored contacts, this person has blonde hair but has colored it pink. You're not going to pick any of those things up. So you've got to recognize these are only predictions, and there are lots of ways, lots of tools and technologies that we use every day to alter our natural appearance. So you've got to take that with a pretty large grain of salt. There are also technical challenges. This kind of technology requires access to a DNA sequencing machine. The typical forensics profile, the STRs that are analyzed against the CODIS database, doesn't require a sequencing machine. It requires what's called a thermocycler. Thermocyclers have been approved and validated in FBI labs for years. DNA sequencers, not so much. So we're just now beginning to see DNA sequencers validated in forensic settings to be used with crime samples. There are ethical challenges here. So if I'm able to look at your genetic information, do you have a right to privacy for that sample? Well, if it's an externally visible characteristic, something that I could pick up just from looking at you, then probably not. But could I also use that sample to learn things about you in terms of health issues? I could. It might be valuable to know that the individual that left this sample behind has a very high likelihood of Parkinson's or has Huntington's. That would give you a better sense of what your potential suspect pool is. But I now know health information, identifiable health information about you. Is that allowable? So there's a real question mark here about, in the interest of trying to solve crime and reduce the likelihood of somebody committing another crime, is that a trade-off that we as a society are willing to take, that I may know this kind of personally identifiable information about you? There are legal issues as well. Almost all countries that wrote laws about the allowable use of DNA in criminal settings wrote those laws before DNA phenotyping. So they were looking at pieces of DNA that were not giving you instructions about identifiable characteristics. So is the use of your DNA for identifiable characteristics going to stand up in court? Are there going to be legal challenges to it? Some countries have gone back and said, yep, molecular profiling is completely allowed. Others haven't touched it yet. The big key, though, is what I've hopefully hit home with you. We have limited scientific knowledge about how likely this genetic information can give me a realistic picture of who you are. Again, it's a genetic prediction. There are a whole bunch of pieces that the science hasn't yet uncovered. We can get close in some instances, but we need to be really mindful that there's a lot of data about the genetics of facial structure and of recognizable features, plus all the environmental factors that may mean that this molecular profile, this snapshot, may or may not be a realistic representation of the individual that actually left the DNA sample behind. Okay, so we've got a couple minutes before I break for cookies. So let me see if we've got any more questions on this one.
How long before health insurance companies want a sample of your DNA?
Ooh, how long before health insurance companies want a sample of your DNA? Okay, I'm going to step away from criminal issues for the moment and into health issues. That's a great question. Right now, we are just at the forefront of beginning to identify characteristics about your long-term health, from your DNA. And right now, if I were to give you your genomic profile, I would be telling you there are some really important pieces in here, but there are some things that you need to take with a pretty big grain of salt because I am missing a lot of information. At the moment, health insurance companies are watching this information really closely and have said that at this point, they don't see genomic information as a useful tool. It's still in the realm of research. It's inching closer to clinical. Work at HudsonAlpha, work in other genomic institutions are beginning to shift us in that direction. And certainly there are reasons when it would make sense to have your genome analyzed. I think we are within a decade of seeing this as a realistic part of your health profile. What about your life insurance profile? You know, it's realistic. Your life insurance companies they make a calculated risk. If you say that you skydive and you drive a motorcycle and you jump off buildings, that's information that's useful to them in calculating your risk. In the same way, if they know from your genetic information that there are things that are in your history, is that allowable for them to use? That's an interesting question for us that we haven't answered yet, and we haven't put any kind of legislation around that either. We've got some protection around health insurance. We don't necessarily around life insurance or disability and long-term care insurance. Long answer to that question. Sorry. Well, isn't that the same as a pre-existing condition? Well, in many of these cases, just because you've got the genetic risk factors doesn't guarantee that you're going to develop it. So a pre-existing condition assumes you've already got the symptoms, you've already got the trait, you've already got the disease. A genetic predisposition says you're at a higher risk, but doesn't necessarily say you will develop it. So I think there's an important trade-off there. Last question before we break. Back to crime.
Okay. You haven't mentioned gender, orientation. You mentioned a little bit about race. And so have those been challenged, like profiling, or is that a portion of the bank?
So gender is certainly in the existing database. The CODIS database, one of the markers is looking at X and Y and gender. So that's already in there. Race has historically not been. We can begin to get some biogeography, some deep ancestry. The caveat, and I think that's valuable, the caveat there is that we tend to think in I was going to say in terms of black and white, but we tend to think that individuals of this race have this kind of appearance. And there's, in fact, huge variation within each different ethnic group. So being able to say they are of this race is less valuable than saying they fall into this like deep ancestry category. But again, there still is some data that we haven't fully fleshed out with that. Okay, so we're going to take a break. We are going to take a 15-minute break, and then when we come back, we're going to talk about how is DNA used for data storage. I'll see you in a few minutes.